My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. We have a good one today. We have Dr. Graham Reynolds, who is a professor of biology at the University of North Carolina, Asheville. And it's interesting to me because, I live literally right down the road from Asheville. I could run over there for dinner if I wanted to and be home at night. But, I haven't interacted with Dr. Reynolds that much. And just in recent times, we're starting to work together on some IUCN, or International Union for the Conservation of Nature, projects around snakes. And I thought he would be a great one to get on the episode, to get on the podcast to talk about IUCN and, all kinds of other interesting projects that he's working on around the world. So welcome to the podcast, Graham. How are you doing today?
I'm well. Thanks, Chris. Thanks for having me. It's great to be here, and I look forward to getting to chat with you.
Yeah, it's great to have you. So I always like to start our episodes and go right back to the beginning, because a lot of people out there, probably think it's kind of strange what we do. So I like to get that trajectory of how we got to the point of being interested in snakes and turtles and these types of animals. So how did that all come to you? Have you always been interested in snakes since you were a child? Is it something that hit you kind of later on as you're going through your career? How did that all come about?
Yeah, I think I think so many of us share that experience of being young people and just love being in the woods and finding critters. And for me, that was certainly part of the path. I grew up on a small farm outside of Asheville, and being outdoors was part of that, and just finding fascination in the natural world. That was the initiation, and everything from reptile coloring books to, getting to go and catch snakes and try to show my parents. That was all part of it. I think that's a familiar experience for most of us. So that's where it all starts. Yeah.
Great. So did you have, being in Asheville in North Carolina, you're in the South, just some incredible snake diversity, but people often don't think of the Appalachians as like this hot spot for snakes. I actually think it's a great place. There's some species that are very interesting to find and learn about, and just some incredible densities of certain species too. So when you were a child there, were you finding many snakes, or were you looking for more salamanders or other groups of animals that were known to have such incredible diversity of?
Yeah, I think I was looking for everything, of course, but I was particularly drawn to snakes and to fish. I really like fish as well. And it's funny. I remember because we could find some snakes pretty regularly, and of course, early on, I found a very dense population of northern water snakes, which was pretty much guaranteed sightings. And so that was always fun. And I remember looking at my field guide to the reptiles of the Carolinas and Virginia, and looking at the maps as a kid, I kept finding that maybe there were more snakes in Virginia than there were around me. And so I had it in my head that I needed to go to Virginia to really see some good snake diversity. And, of course, like looking back on that, I find that kind of silly, but I think probably what I was keyed into was maybe that confluence of the coastal plain area up around, maybe Virginia Beach. It's hard to know what a 6-7-year-old is thinking, but yeah, I think I did start to appreciate that we had some pretty significant diversity not too far from here in Asheville. And, not just we've got some good snake diversity, but certainly salamanders. It is such a center of diversity for salamanders that you can't help but appreciate that.
Yeah. And you mentioned fishes too, just incredible diversity of fishes and invertebrates. And it's definitely a hot spot for biodiversity in the world. But, so you're growing up in this beautiful region and, pretty rural, and you said you're on a farm. When did it hit you? When you're out there, kind of enjoying the natural world, kind of exploring. When did it hit you that like, oh wait, this is people do this for a job? Like this is something that I could pursue a career in. Did that happen early on as well, or was that something that hit you later?
That was fairly early on. I think it was around sixth grade. I started to have this inkling of an idea that, people could do these kinds of things that I enjoy as part of their career. And, where it went in that part of middle school was thinking, I really liked going to pet stores, in particular aquarium shops. And so started thinking, well, maybe there's something in like, husbandry there. But anyway, it was the ninth grade when I decided I wanted to be a college biology professor. That's when it really coalesced for me. And, that's a bit unusual. I'm not sure that many of us who are, academics really might have settled on that early. But for me, it really did happen in the ninth grade. It just kind of all came together. This idea that not only could I be a biologist and work on these amazing critters, but also be a teacher and a scientist.
Wow, that's amazing. I think in ninth grade, I probably wanted to be a pro football player or an astronaut or something. So that's pretty amazing. but
that would have been a good path to
us. Anyways, great. So you kind of knew this kind of early on, and obviously I mentioned you're at University of North Carolina, Asheville. So you're close to home, or at least where you grew up. So how did you, what got you from point A to point B? Meaning, did you decide to stay home? Like when you were going to do an undergrad, were you like, I'm going to stay close to home, this is the place I love? Or did you end up kind of going off and going to universities and other places? And how did that all happen?
Yeah, well, I mean, it's funny because I'm really more of a tropical biologist, and that sort of started to happen for me. I did an internship my junior year of high school. I actually finished all my classes early that year and took my exams a month early and went and did an internship in Mexico. And that was transformative. I mean, I really, for whatever reason, despite growing up in such a biodiverse region, I was always drawn to the tropics. And so getting to go and work in tropical dry forest in central Mexico and the Pacific coast, it just really opened my eyes to what all was possible. And I ended up being an intern for the next 3 years on that project. But what it made me do was really want to kind of expand out a little bit and start to explore what options were out there. So I started looking in particular at programs that might have, a marine component. I was really into marine systems, not just the fishes, but maybe islands and things like that. So that's how I selected the universities I wanted to go to, and eventually chose Duke. Not too far away, but they had a great biology program and they had a marine station as well. So ended up there.
Oh, okay. Yeah, one of my kids is actually looking at Duke for the same reasons, marine related interests. So,
yeah.
So that's great. So you go to Duke, and on top of that, Duke is just, in terms of ecology on a global scale, I think of Duke as a great place for all of that. So, as an undergrad, did you have good opportunities to say go out to the marine station there and continue working in the tropics, or was that something you had to kind of hunker down and focus on the Piedmont of North Carolina?
Yeah, well, I spent a lot of time in the Piedmont. I love the River State Park. I spent a lot of time out there as an undergraduate. Of course, the Duke Forest too is a great resource. There's actually some great herps in the Duke Forest. But I got to study at the Marine Lab. That was amazing. I actually got very interested in marine invertebrates, believe it or not, taking classes out there. You couldn't help but just get excited about that kind of stuff when you're right there, digging through the mud and learning about all these things. Parchment tube worms, they bioluminesce inside their tubes. Like, why would they do that? You can't even see them. Anyway, just stuff like that got me interested. But I joined a lab, and it was Dr. Susan Albert's lab, and she's still there. And she's a primatologist. And I was interested in animal behavior at the time. And I took her animal behavior class, and part of that was we had to write a grant proposal as one of our assignments. And so, I had done a study abroad in Costa Rica and Panama and got, even more interested in the tropics. And when I took that class, I said, well, could I write a proposal that I actually submit? And Dr. Albert said, yeah, sure, why not? I think she thought like, okay, yeah. But I wrote a proposal to study poison dart frogs on islands off the coast of Panama. And based on some research from Kyle Summers, who is at East Carolina, and I read some of his work. And so I wrote the proposal and sure enough got it funded. And so Dr. Alberts was kind enough to let me go and do that as my undergraduate research. And that's when things really started to materialize in terms of what I knew I wanted to do, which was herps on tropical islands.
Yeah. So what aspect of the, kind of this first island experience with herps, what aspect of the poison dart frogs were you interested in?
I was looking at mate choice. So Kyle Summers had published a paper in 1999 that really showed that these frogs, these are strawberry dart frogs or blue jeans dart frogs, that there's all these different color morphs on these islands off of Panama, and each island has its own color. They're all the same species, and their male songs are similar, but the coloration's so different. And these are very visual frogs, right? So they have this whole courtship where the female's really visually assessing the male. And so for that reason, the females are choosy. And Kyle Summers showed that the choosiness was in part due to their color. And because some of these populations, the frogs are red, some are green, some are yellow, some are blue. And so I wanted to extend that and look to see if there was any indication of whether females could sense color patterns, because some frogs have big black spots, some have no spots, some little spots. So that was the project, was to extend what Kyle worked on and really see if color and pattern were involved in mate choice. And that's what I was doing, was essentially these behavioral trials to see if females could discern males from their own population or not, based simply on patterns. So I used these color filters to make sure the light was all the same color, and then they could still see the contrast.
Wow, that's fascinating. I've seen some poison dart frogs over the years, working in the tropics, but I know very little about them. I guess I would have assumed that the bright color patterns were more kind of like warning patterns. I guess I never thought that some of it might have to do with like sexual selection and other things. So you think, is it a combination thereof? You think in many species, kind of like a aposematic, like a warning type color, or do you think it's mostly sexual selection?
Yeah, great. So it's definitely a warning color. It's aposematism. I mean, these are chemically defended frogs, and they are diurnal, and they're kind of carefree. When you see them in the forest, they walk around, they call. They're not trying to hide, right? So that's the classic, sort of aposematism. But the fact that the females use this as a sexual signal, so that we really put that forward as it was one of the first examples of what's called a magic trait. And these are phenotypic traits that are involved in both natural and sexual selection simultaneously. And the reason that that's interesting is it had been predicted through some mathematical models that would cause really rapid evolution. If you had both natural and sexual selection acting on a trait, you should expect to see rapid changes in that trait on an evolutionary timescale. And so we really think that's a good example of a magic trait. This sort of what at the time was a hypothetical prediction, but we showed that this is a good example of an empirical magic trait. And so part of the reason we think that all these islands, despite having been, connected relatively recently, why the frogs look so different is because you see this really rapid evolution of sort of moving towards a single color and pattern on each island, because there's so much selection to narrow that down, right? If you're thinking natural selection, then you need to make sure you're presenting a consistent color and pattern to potential predators as a warning color. Well, if you're presenting to females, you need to be presenting consistency as well. So you get a pretty rapid narrowing down of any variation, and it happens in different ways in different islands. And so you end up with this diversity of colors, despite being all the same species. So it's a pretty fascinating system. And we contributed a small part of that, but just thinking about how the females were a part of that.
Yeah, that is fascinating. And we haven't even left your undergrad yet. So,
No.
yeah, I mean, that's actually, I joke about that, but that's actually a good, for any of you students out there. And I did the exact same thing as Graham. Like, if this is what you want to do, you have to go all in, in your summers, like Graham was talking about in Mexico, you do internships and, jobs that are related to the field and take every opportunity to do anything you can undergraduate research on. I mean, it's such a competitive world and in this space because so many people want to do it, and you're hearing one of the ways to make yourself stand out. But anyway,
Yeah.
so you're getting this interest in herps is growing, and the tropical interest is growing, and now you've brought in this island component, and you're still in your undergrad. So did you take those interests and kind of bring those into graduate school with you? Where did you go to grad school? Did you do a master's, just a PhD, both? And what'd you work on,
Yeah.
for those degrees?
Yeah, I definitely wanted to continue the tropical island theme. I actually initially wanted to work with Ted Case, who's a really well-known, um, you know, island biologist, but he was retiring at the time that I was applying to grad school. But he had worked in places like the islands in the Gulf of California, for example. So anyway, I knew that I needed some more training in particular population genetics. That was something that I was really getting interested in, and I had not had a lot of training in that. Just one class, I think, as an undergrad. So I started looking around, and I found that there was this group of, at the time, relatively younger faculty coming out of UC Davis who were taking up faculty jobs at the University of Tennessee. And they were seeming to put together a pretty interesting program. And so I applied to Tennessee. And and I remember my interview weekend, I met these people and I was just really amazed at the work they were doing and the ideas they had. And I knew that was the kind of place I wanted to be. So that's where I ended up, was University of Tennessee, straight into the PhD program. And, right away started taking some intense classes in learning some computer languages like SAS and R, and then some higher level population genetics. And that first year was tough. Yeah. For those students out there, just know, it's hang in there.
Uh-huh. So as part of your dissertation program there, did you come in and kind of fit into a larger existing program where they wanted you to do X, Y, and Z, or or were you able to kind of develop any ideas you wanted around the dissertation? How did that play out, and how did you end up? Basically, I'm asking, how did you end up getting towards your dissertation project?
Yeah, well, I mean, I was lucky. So I joined the lab of Ben Fitzpatrick, and Ben was already well known as somebody who was a really deep thinker about some key ideas in speciation and population genetics and hybridization. He had been working on the California tiger salamander complex, which is a fascinating example. But I remember asking him, I said I would like to kind of develop some island projects, and he said, yeah, sure. And that was the beauty of joining a new lab. He had just started at University of Tennessee, so he was kind of open to ideas about different ways in which the lab could be doing different kinds of research. So that really appealed to me. And then I remember we had some other folks there. Dan Semberloff is at Tennessee, and Sandy Echternach was there. And Sandy was a herpetologist and worked a lot in Central America and Latin America. And Dan, of course, well known for working on islands in the Florida Keys and other places. And so I remember I pulled Sandy aside at one point. I said, Sandy, I need to find a group of islands that has a certain set of characteristics. I wanted big islands, small islands, islands far away, islands near. And so we kind of spun the globe together. I really wanted to work in the Gulf of California on rattlesnakes. That was what I wanted to do. And luckily, some of those folks, including Ben and Sandy, kind of said, well, maybe that's not such a good idea. It's like, they were concerned about some of the risks of that, perhaps. But so we landed on the Turks and Caicos Islands, and a couple of reasons. One, it's not that far really to get there. It's at the southern end of what we call the Lucayan Archipelago, which is the Bahamas and Turks and Caicos before you get to Hispaniola. And we had some former grads from that program at Tennessee who were working there, including Glenn Gerber. So we had an in already, and we had some knowledge, and it fit the bill in terms of what we wanted. And what I wanted to do was to sort of build on some ideas about conservation and population genetics, in particular as they relate to island archipelagos. So we started, that project. My first chapter was a theoretical chapter about, trying to simulate some data and see if any of these ways that we go about finding, for example, genetic bottlenecks hold up. Turns out the program bottleneck that everybody was using is subject to type 2 error, like a lot of it, if there's population structure. So that was something we found.
Explain for the audience, because not everybody who listens is a scientist. Like, how would you describe type 2 error to just, to your child or something?
The idea was, so this program was meant to be able to, you were meant to be able to use genetic data to be able to detect the presence of a bottleneck by looking for essentially a mismatch of rare to common alleles. So the idea is that when you have a genetic bottleneck, you should lose a really high proportion of rare alleles. And that's just because you're reducing the number of individuals in the population, and rare alleles are going to be in fewer individuals. And so this is the kind of pattern that you would expect to see. So that program is sort of trying to detect that pattern. And what we found is that you can preserve rare alleles if you have population structure. So if you're, for example, a metapopulation, so if you're a bunch of islands that really constitute a population that has some kind of connection by gene flow, that you actually can disguise that signature, or you can amplify that signature. So the problem is that it comes in when you try to use this program to detect bottlenecks. You might detect bottlenecks simply because you're seeing a mismatch of what you'd expect in terms of the frequency of some alleles versus others. And that's a real problem because most populations are not going to fit sort of your classic population bottleneck model where you've got one population that's all connected together, and then all of a sudden the population gets really reduced. And that's what you're looking for in terms of signatures. But if there's anything else happening, like genetic drift or gene flow or even extinction and recolonization, then you're going to get either detection or no detection of bottlenecks when there were or were not. And so you're just not getting a good signal there. And that's a real problem. And we found that a lot of these supposed bottlenecks that have been detected were probably not genetic bottlenecks in reality.
And so which species were you working on in the Turks and Caicos?
So then we wanted to take that out to a more empirical approach. So I worked on curly-tailed lizards, which are this really cool group of critters. At the time, I picked them because they're really abundant on these islands. And I could have picked the nulls, that would have been an obvious choice, but I wanted to work on a lizard that we didn't know that much about. Mm-hmm. So they are great.
Kind of snakes with legs for the no. I know you're into taxonomy, so you're probably like cringing right now. But anyways, so so what did you? So you said you were looking at some of these empirical example of some of this. So what did you end up finding in the dissertation work?
Hey everybody, I just wanted to take a break to let you know that you can stay connected to the Orian Society by signing up for our monthly newsletter. Be the first to receive exciting updates on reptile and amphibian conservation, from the latest news and staff blog posts to exclusive announcements like upcoming Snake Talk podcast guests. Go to our website at www.orian.org and sign up for the newsletter at the bottom of our homepage.
One of the things we found, there were all these subspecies of curly-tailed lizards described from that archipelago. We found that those subspecies didn't really match the distribution of genetic diversity and divergence, which that wasn't surprising. We also found
meaning that they were all one species, or just that okay, gotcha.
Yeah, they're all one species, certainly, at least in the Turks and Caicos. And these islands that had subspecies described, they weren't really distinct. And the morphology even that was used to describe them was more variable than was appreciated at the time.
Okay.
So, so there was a mismatch there in terms of what they were called and what they probably actually are. Again, not surprising. That was pretty straightforward. But we also found that the kinds of data that we were collecting weren't really going to work for the kinds of things we were doing. Plus, we discovered that the methods we were going to use to try to estimate, some of these, what we call population or demographic dynamics, like bottlenecks, genetic drift, extinction recolonization. The program bottleneck didn't work for that. And a couple of the new methods that had been published recently at the time didn't work either. There was one called decomposed pairwise regression that didn't work. So we found that there just weren't very many good methods to reconstruct, demographics in these archipelagos. And so happily, there have now since been new methods developed, including something we call approximate Bayesian computation, which helps a lot with this kind of thing. But it's a complicated thing. And I think what we were trying to show at the time was that it's more complicated than people were willing to admit. Mm-hmm. And that whole idea about trying to understand the importance of bottlenecks, especially as it relates to species extinction, that's something that I'm still continuing to work on, because we still misrepresent the importance of a population or a genetic bottleneck in the context of species extinction. And so this is still a theme that's part of the research that I'm doing.
Okay, interesting. Great. So that's your dissertation work, and let's kind of close the loop on how you got to where you are now, and then I want to dive a little deeper into some of this stuff that you're working on. So, but you've been building over time. There's an interest in herpetology, we'll say reptiles and amphibians. There's an interest that you develop in the tropics, and then you develop this interest in islands, and you add in the kind of popular, you have the interest in behavior, but then you add in the population genetic component. So you're really building these pieces over time, which have all obviously shaped what you do today. So you finished the PhD. How did you go from University of Tennessee to UNC Asheville? Is that something that happened at that point, or did you postdoc, move around? What was the trajectory there?
Well, yeah, there's one missing piece on that dissertation. That was my third chapter, which was on boas. And the way this happened was rather serendipitous, because in working on these islands in the Turks and Caicos, I started to hear that there were boas on some of them, and we didn't know anything about these boas really, just a little bit about them. But in talking with Glenn Gerber, we sort of realized that one of the islands had probably a dense population of boas. And so I pitched an idea to Glenn, because I've been interested in boas since I was a kid. When I was young, I think maybe 11 or 12, we were in the Bahamas with my family, and I saw a Bahamas boa that had been killed by a cat. And at that time, I remember coming back. This was before the internet. And so looking in encyclopedias, trying to learn about these boas, and just realizing there just wasn't that much known or written about them. And so, the opportunity to maybe work on boas a little bit while I was there just was irresistible. So I pitched Glenn this idea. I was going to go for 2 weeks to this island and just see if there was a feasible boa project to be done, right? Like several people had said, don't bother, you're not going to catch enough of them to make a dissertation chapter. Well, I caught 50 in that first 2 weeks. And so I was like, well, we got something here. So that project is still going on. Every year we go and work on this population. We've pit tagged thousands of boas in that population now.
And so you're doing like a mark recapture,
that's right.
Measuring, taking, all kinds of information on individual animals. And so is that on one island in particular, or are you doing this on multiple islands?
We've done it on multiple, but there's one island that's our core island. It's called Big Ambergris Key, and it's a private island. And we're, part of what we do there is work with the island owners to ensure good stewardship. So keeping an eye on the populations there's also iguanas there and some other interesting critters. So it's been a good partnership, and we've managed to learn a lot about the boas now.
Yeah. Okay. Well, I want to come back to the boas since that is a long-term project. So, but let's put a bookmark there, because I want to hear how you got to UNC, and then I want to just talk a little bit about islands and species and snakes in general, and then we'll go right to the boas. So, but tell me how you got to UNC Asheville.
Yeah, well, so I knew I wanted to be an academic, so a postdoc was going to be required, a postdoctoral fellowship. That's very typical for academics to do that before they take a faculty job. So I applied for a number of them, interviewed for some. I was looking at some like with the Smithsonian and various places, and I ended up connecting with Liam Revel, who had just started a faculty position at University of Massachusetts, Boston. And I was particularly drawn to Liam's lab because he was looking for somebody who was a tropical herpetologist. He really wanted to lean into that side of the things he was doing. He works on anoles, but Liam's also really well known for a lot of the tools that he's developed to study phylogenies in R, basically the software R. And so it was a really good arrangement because I knew, and by the way, for the students who might listen, a lot of what I've tried to tell my students is it's all about the tools in your tool belt, right? Like continuing to add tools. That's how I've always viewed my career, and how I think, I want my students to view it is, just think about what are the things you'd want to learn and get after those. So I knew I wanted to learn more about phylogenies and phylogenetics. So for me, it was a good match with Liam. And Liam wanted to do some projects in Puerto Rico. And so that was a good match for him because I could do that. So that was a great experience. So I spent 2.5 years as a postdoc with Liam, helping him get his lab going. And I learned a ton. And, he's been an amazing mentor. And started getting more and more interested in anoles as well as boas. And so then I moved over and joined Jonathan's lab, Jonathan Lassos, who was at Harvard at the time. So I moved to Harvard for 2 years and did a postdoc there. And that one was great. And I learned a lot there because I learned about managing a lab. Because not long after I got there, Jonathan left to do a sabbatical. So I ran his lab there for about a year while he was writing a book. And I learned a lot about what that meant to mentor grad students. And so it was a great opportunity. And Jonathan has been an amazing mentor as well. So I've really been lucky. That's part of it, is I've been very lucky with the folks that I've gotten to work with and learn from. Mm-hmm. But when you're a postdoc, what you're doing is looking for your permanent job. Postdoc's a great job, but it's temporary. So I started looking around and thinking hard about what kind of job I wanted. And this is something that students are going to wrestle with: is do you want to lean towards an R1 school, a really research-oriented school like where I was at Harvard, or do you want to be more on a school where you can be rewarded for teaching and things like that, and at a more maybe smaller classes and things like that. So I've always been interested in teaching, and the opportunity came up to start to apply to some schools where teaching was a big focus, and I got more and more interested in that. And sure enough, a job in my hometown opened up looking for a vertebrate zoologist. And so it was almost too good to be true. So that's what got me back to Asheville, where I'm from and where I've been since.
Yeah. How much was Asheville a draw? Like, were you trying to get home, or if that same job came up in Iowa, would you have been just as happy?
Yeah, I applied all around, and don't get me wrong, I loved Boston. I would have stayed in Boston. I think that's a great city. So I was looking in upstate New York. I was looking in a lot of places that had those kinds of schools where, I could really teach undergraduates, mentor undergraduate research, which is something that I really benefited from. And I wanted to be able to offer that to students. But a job in your hometown, come on, that doesn't happen very much in academia. So that was a big part of the draw. And, I came and interviewed, and I really liked the folks in the department. So that was great. And it's been a really good fit.
Yeah. Excellent.
Yeah.
Great. Well, I went to UMass as well, by the way, for a couple of years.
Nice.
In Amherst, but
I
grew up in that general area as well. So anyways, so let's shift a little bit and talk about islands. A lot of people out there, they probably love to go sit on a beach and, have a nice island cocktail. But I don't think many people think about islands in terms of nature, in terms of snakes, and how there's so many superlatives about ecology that have to do with islands, conservation, so on. If you could just kind of give us a very high level overview of why you think islands are important, both, say ecologically, what makes them somewhat interesting or unique, from a conservation perspective, just sell us on islands. And then I also want to get into kind of snakes and islands.
Yeah, well, it's a pretty easy sell when you start looking at it. We often say, we use this sort of phrase that islands are laboratories of ecology and evolution. And the reason we say that is because they really do offer us as biologists a lot of insight into the processes and mechanisms that yield these biodiversity outcomes, be they ecological or evolutionary outcomes. And the reason for that is because, oftentimes we see these kinds of things manifest in a way that's more clear or more quickly than they might manifest, say, in a mainland type system. And, this goes back well before Darwin, but this has been appreciated for a long time. But islands offer us that ability to really see these kinds of outcomes. It's almost like, what happens when you might, start afresh with a new ecosystem and let things run and see where you end up. That's the kind of thing you get with islands around the world. And so in that way, they are sort of these little microcosms or mini experiments in evolution and ecology. And they are so powerful for teaching us what we now know about those mechanisms because they allow us to uncover what it is that's led to that particular outcome. So, in that way, they're of immense value to us as biologists. Mm-hmm. But for me in particular, being interested in population genetics early on, islands are great because they're these little, discrete units. And so those fit into our models a little bit better. When you've got units with discrete boundaries, then you can start to use those as models in a little more, a little easier way, I would say. There's also something, what's that?
Oh, I was going to move into like the conservation side of islands. Is that where you were going, or
that's where I was going? Yeah. So
I was just going to say, there's another side of the coin that islands, species on islands are also some of the most threatened in the world. So maybe you could touch on that a little bit for folks.
Absolutely, yeah. So islands oftentimes produce these astounding biodiversity outcomes, but they are oftentimes very sensitive, right? They're sensitive to all kinds of ecological change that might befall them. And so what we see is rates of extinction on islands being much, much higher than rates in mainland type situations. And so, from a conservation standpoint, there is significant concern about loss of island biodiversity. Again, like you can imagine all of these little natural experiments that have played out and yielded all these biodiversity outcomes. And then we're starting to lose those, right? So that feels like a very pressing concern. And so it is something that a lot of us think hard about and lose sleep over.
Yeah. And what are some of the major, I could run through some of them, but let's hear from the expert here. What are the, if we're talking on a global scale, there's a number of factors that, are especially impacting island species really globally. And so I'm just curious if you could kind of go through some of that for listeners so they could understand how this loss of biodiversity is what's driving it, basically.
Yeah. I mean, it's people, right? I mean, so, there's a couple of ways. One is if you can go back even thousands of years and you find that humans have a really outsized impact on island wildlife. Some classic examples of that are the loss of giant birds in Madagascar and New Zealand, the elephant birds and the moas, respectively. So those were probably pushed towards extinction by humans arriving to those areas. We tend to have a big impact when we arrive in island ecosystems. And it's not just us and what we eat, but it's also what we bring with us. So invasive species are another huge factor in island extinctions. And so, you look to a place like the Mascarenes in the southern Indian Ocean that's unfortunately an area like, Mauritius and Réunion that are known for extinctions of a lot of really iconic terrestrial vertebrates, like the dodo. And we can directly draw a line between human impacts, including the things that we like to eat and the animals we bring with us, and the loss of a lot of that endemic biodiversity. So, islands tend to be insulated from a lot of the perturbations of the world. And then when humans start to arrive there, we often have a pretty dramatic and fast impact on those ecosystems.
Yeah, one of the projects that Orianne is very involved in, really our only island-based work is in the Indian Ocean, not far from Mauritius and some of these places, but we're
Mm.
doing a lot of rewilding work for giant tortoises throughout the Seychelles and those areas. And just, they've been gone from so many of those islands for so long. But anyways, that's a side note. So let's talk about snakes and islands. Like, how, if I just give you those two words, snakes living on islands, and you're thinking kind of globally, or you could think kind of tropically around the world. How do you think of, biogeography, patterns of distribution, general ecological patterns, speciation, conservation related issues? Like, how would you at the highest level kind of summarize snakes and islands?
Yeah, I love it. One of the amazing things about that is when you look across the snake fauna on different islands, a couple of things emerge. One is that you've got some snake species that seem to be able to colonize all different kinds of islands in all different kinds of contexts. And this ability, it's something that is perhaps intrinsic to some of these snake species, their ability to adapt in a generalist way to novel environments, their ability to colonize these islands either on their own or in some cases assisted by people. And so that's an interesting outcome. But then there's also this outcome where we see a lot of endemic biodiversity of snakes. And so, for example, the genus I work on, Cheilobothris, is an entirely insular genus. It's only found on islands, right? So it's a product of a dispersal event from South America to the Caribbean in the Miocene that has now led to all of these 14 species in that genus. And so we see some radiations like that that are pretty exceptional. And then the third part of that is abundance. There are some islands where the snakes are just ridiculously abundant, way more abundant than they might be in any mainland situation. And so, that happens with both species that are found on both the mainland and the islands. There might be islands where there are way more snakes on the island. For example, Boa imperator, the Central American boa. Good luck finding one of those in, say, the rainforest of Honduras. It's going to take you a while, I know from experience, to find one. But there's islands off the coast where it's just lousy with them, and you can find 20 in a night. So there's an abundance thing that's happening on islands. And so that, for me, those three things together, the fact that some snakes are found on a bunch of islands, but then you've got some endemic snakes that are a product of having their ancestors having colonized the island, and then this outsized abundance, that just draws me to them.
How about, are there certain, you talk about snakes being, able to come onto islands and, kind of acclimate and be generalists and good at that as a taxonomic group. Are there certain groups of snakes that have been, boas, colubrids of some type that have been especially good at colonizing islands, or is it pretty well represented across the taxonomic groups?
Yeah, there's definitely some that are way more common. Some of the nature scenes in different parts of the world, boas especially. Boas are quite good at this, and a whole lot of boa species. We recognize 66 boa species, by the way, and a bunch of them are found on islands, either exclusively or in part. So boas do seem to be quite good, and this is circumglobal. We have boas in the South Pacific. There's a genus of boas called Candoya that's found from Halmahera, which is in the eastern part of Indonesia, through New Guinea, all the way out to Samoa. And that is a distribution that's just astounding. And that's just really speaks to their ability to colonize across ocean boundaries. That's a pretty amazing example.
Yeah, and if you had to speculate how most of this dispersal is happening, are these snakes floating on debris? How do you think a lot of these snakes are getting around the world? Across the Pacific Ocean, like you're talking about.
Yeah, so some of them certainly are capable of floating. We actually have some very interesting historical anecdotes. There was a priest or somebody in the Lesser Antilles on St. Lucia who actually observed a boa constrictor coming ashore that presumably had washed off of some other island or off the mainland of South America that came ashore. And he speculated in concern about what might happen to the sheep that were on the island.
Poor sheep.
Yeah, exactly. So,
are there boas on St. Lucia?
Yeah, there are. Yeah.
So I thought, okay.
Yeah. So,
and there's also the St. Lucia's one of the small number of islands in the Caribbean that has a viper on it too. So
yes, indeed. Yeah, there's only two islands that have pit vipers, at least in the Caribbean proper.
Yeah.
Both in Western Caribbean.
There is a rattlesnake, a Dorissus group that comes. It's on islands, but just off the South American coast. Is that right?
That's right. Yeah, there's the famous one would be the one on Aruba.
Aruba, but there's another that I was thinking of. Sorry, go ahead.
Yeah, I think a couple of islands off Venezuela might have or have had.
A population of those. But
and then of course the islands in the Gulf of California. I mean, you have a rattleless rattlesnake there. But amazing.
So, snakes on islands. Let's talk a little bit more about these boas and the Turks and Caicos. So, I guess first, describe them. They came from, South America, as you were talking about biogeographically. So, do they look like boas from the mainland? Do they look different? Like, what's their appearance, first of all?
Hey everybody, I just want to take a quick break and tell you that the Oriane Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.oriane.org to learn more about our work and how you can get involved.
Yeah, they don't look like boas. Many of them look like they're kind of the size and shape of a large garter snake. Like these are small boas. So the interesting thing here is this radiation of Chilobothrus, there's 14 species. When I started working on it, we recognized only 9. So, we've added to that species number a couple of ways, both in terms of just in situ discovery of new species, but also recognizing some that are distinct lineages from, it's called splitting, but it's essentially what we thought was one species. But it's amazing thing about this whole genus is that there's kind of two flavors. There's big boas and small boas. And the big boas, some of them are absolutely enormous, even by boa standards. So, for example, the Cuban boa can reach lengths of 4 or 5 meters, perhaps. And that is a truly enormous snake. And that's a species that it's the only boa that's found on Cuba. And they're huge. You've got other large ones. The Jamaican boa is large. There's one in Hispaniola. But there's also small boas. And some of these are really, really small. So the smallest one in this genus is also found on Hispaniola. And that's the Hispaniolan vine boa. It's actually one that we just discovered in 2020. And those are less than half a meter snout vent length. And they look a lot like some of the neotropical vine snakes, the really small ones. We call them a blunt-headed vine snake. It's Hymentodes is the genus, but they look just like that. So the small species tend to be specialists in terms of being mostly arboreal and feeding on just lizards or frogs, whereas the large boas are generalists. They live on the ground, in the canopy, everywhere, and they eat everything. And so there's this interesting split in that way. And what we discovered was that it's not the case that all of the big boas are each other's closest relatives and all of the small ones are each other's closest relatives. Instead, each of these has evolved independently over and over again in situ on different islands of the Greater Antilles. So, for example, Hispaniola has 1 large generalist and 3 small specialists. That is a unique radiation for Hispaniola. And those small boas on Hispaniola are not the closest relatives of the small boas on, say, Puerto Rico.
That's really cool.
Yeah. So it's independent evolution and it's convergent evolution on each of these island groups. And so that's a pretty fascinating outcome.
And do you have any estimates to, how far back in time did some of these species radiate out from each other?
Yeah, most of these ancestors that would have colonized what at the time were the Proto-Antilles, this would have been in the late Miocene is when a lot of this was going on. And then a lot of the in situ speciation happening in the Pliocene and even into the Pleistocene. So, within the last 5 million years. So, yeah.
So the species that you're working on, the Turks and Caicos, where you have the long-term mark recapture work, describe that species to us. Is it one of these?
They are so cool. They're like the gentlest snakes. You walk up to them and pick them up, and they just drape. They're amazing. But they only get to about 1.5 meters, so they're pretty small. And like I said, most of them are about the size and shape of a garter snake, but they have really dramatic sexual size dimorphism. So the males are very small, never more than about 700 millimeters snout vent length. But the females can get up to 1.5 meters and weigh up to 2 or 3 kilograms. So you put a male and a female next to each other, and they don't even look like the same species, full-grown male and female. So that's a really amazing thing about them. And they have these 2 distinct color patterns. There's a striped morph and a spotted morph. And it's one of the few, if only, boas that really has a distinct color dimorphism in that way.
But it's not specific to the sexes. You could have a striped or a spotted female and vice versa. Yeah.
That's right. Yeah. It's a pretty unusual thing. And unlike garter snakes, where that's a little more, worked out why that's happening, we don't know why this is happening in these boas. We don't have good explanation that might have to do with natural selection or anything to suggest why some are striped and some are spotted. It's probably just differences in the way the melanin is deposited, but it doesn't seem to be under strong selection.
So what have you, like you said, you've been working with this species for quite some time. So what have you learned about their ecology over the years?
Yeah, I mean, one of the things we try to do is just figure out where they are. So, finding new populations and how many there are. Our newest population we discovered just a few years ago, actually. So we're still finding new ones.
So do they live, do they do well around people? Do they live in relatively untouched forests? So where do they live?
They are very sensitive to people. We can still find them on the really, heavily inhabited islands, but they are so defenseless. Like I said, they're gentle. They're fragile. I mean, we find a lot of them killed by cars. We find them, people, of course, just they kill snakes all the time. I don't have to tell you that, but their pets, cats eat them. Rats will eat the babies. It's kind of a mess out there when people start getting to a certain density on these islands. So, islands that are ecologically intact, these snakes are occurring at really high densities. So the island I work on is 400 hectares, and we estimate population sizes of up to 10,000 snakes in that island. So that's a very high density.
A lot, yeah.
Yeah, we don't see densities like that anywhere else, especially islands that have a lot of people on them. So one of the reasons is, the other reason is food. These are saurophagous, so they eat lizards mostly. They'll eat some birds, but mostly lizard eating. And one of the first lizards to go on islands where people go is the iguana, the endemic rock iguana. Well, this is an important food resource for the boas, especially the juvenile, the hatchling iguanas, and the boas, when they're females, can get big enough to eat adult female iguanas. And so when that food resource goes away, then probably impacts the boas significantly. So the islands where we find robust boa populations also have iguanas.
Gotcha.
Yeah.
So any other kind of interesting ecological things you've learned? You do a mark recapture, you've been able to, you talked about the densities of populations in some areas, but anything else you've learned about, like, say, survival or anything you've been able to estimate from those mark recapture data?
Yeah, we've done mark recapture and we've done radio tracking, radio telemetry. So the mark recapture suggested to us that the boas really don't move that far. And that was borne out by the radio tracking, but not in the way we expected. So, for example, boas tend to use not necessarily the same refugium, place where they hide during the day. These are nocturnal, but more or less the same area within a few meters. And so we kind of thought, well, maybe they just kind of hang out in that area and that's really just where they are. But with the radio tracking, we found that that's not the case. They actually go out on these long foraging missions and then come back to the same spot. So we were tracking one female that she was hanging around the same area, and then all of a sudden she wasn't. And so we had to go off and find her. And just that evening, she had moved that far. We found her way out in the mangroves. I had to wade through the seawater to get out to where she was, waist deep. I had to wait for a black-tip shark to pass in front of me before I could get out to where this boa was out foraging. And then, sure enough, in the morning, she came back. So they'll do these long foraging bouts in the night and come back to more or less the same spot. So they do actually move a lot more than we thought, but just not changing locations a lot.
Yeah, so if you're studying these animals and you're just getting daytime locations for them, you're missing a huge part of what they do. So
exactly, yeah, it was fascinating. And habitat, we didn't know they used mangroves.
Yeah, that is interesting.
Yeah.
So, I mean, we can talk about this. I mean, you talked about this island a little bit, but let's talk about the group in general. All those boas throughout that region. You talked about islands and snakes, and we talked about conservation and biodiversity on islands in general, and with snakes. Talk about these boas throughout the Caribbean there, and what's kind of their conservation status in general? How are they doing around all these kind of invasive, exotic, say, predators and other things of that nature? Do some of them do really well around people and things like rats and whatever it might be, or are they all generally kind of declining as a result?
Yeah, great question. So it's both. So some species we have seen, for example, the Hispaniolan striped boa and the Puerto Rican boa, they seem to be doing well, if not better than in the past. And that's probably in part because they have switched to eating rats. And so they are capable of living around humans pretty well. And in fact, our genetic data for the Puerto Rican boa really show that population has expanded tremendously over the last century. As Puerto Rico has been reforested after, having all the forest cut down for sugarcane and coffee, the island is getting much greener, and the boas have recovered significantly. Now, other boa populations are doing really badly. So the Jamaican boa is doing very poorly, in part because of persecution, people killing them. But in this genus, we also have the world's most endangered boa, and that's the silver boa. And this is one that we discovered on an island in the Bahamas, an uninhabited island, in 2015. So we weren't looking for them. We found them and discovered this new species. And there are fewer than 150 total.
Wow.
For the whole species.
So and you call them a silver boa? Do they actually look different than other boas? There's a genetic difference you found? Are they actually silver?
They are actually silver. We named them silver boa for two reasons. One, the first one was in a silver palm, but they are also silver colored. They are really spectacular. Yeah. And we're very worried about them with that few individuals in terms of the whole census population for the species being less than 150. And we're finding no genetic diversity. So we have some major concerns about that species. And that's one that keeps us up at night a little bit.
Yeah.
And yeah, they are astounding. Look up some photos and they are just incredible. I encourage the viewers to check those out.
Yeah, I need to look at a picture because just based on the name and how you describe them, I'm like, this is probably a species that is, would be of interest to the black market, pet trades, those types of things. So
we're worried about that. So we've been careful about, talking about where they're actually found. We're more general about it.
Yeah, good. Well, we've been going almost an hour. I want to ask you one more question, then we'll kind of begin taking this episode in for a landing here. But the last question also relates to conservation, and both you and I have been involved with IUCN. I mentioned that in the intro. I founded the Viper Specialist Group, and you've been involved with different specialist groups, and we're now both involved on the same specialist group. But why don't you talk a little bit? I've done episodes on IUCN and specialist groups, so you don't need to give that broad background. But why don't you talk a little bit about your work and how you ended up becoming involved with IUCN and what you're doing with IUCN today?
Yeah. So, obviously having been interested in conservation since grad school, but really, I guess what started it was my colleague and friend, Alberto Puente Rolón in Puerto Rico. He and I decided that we wanted to have a Caribbean and Latin American BOA group. And so we branched off the BOA specialist group at the time. It was an IUCN BOA and Python specialist group. to start this Caribbean and Latin American boa group. So that's really where I got involved, started getting involved with IUCN. We hosted a meeting in Puerto Rico, and that was great. So then I started working more with IUCN, the boa and python specialist group in particular, and then also got involved with the anoline lizard specialist group. And I started getting especially interested in the red listing process. So I participated in some red list workshops in the Caribbean, and then got really interested. And so when the snake specialist group was being formed, talking with Dan and some of the leadership there, just mentioned that red listing is something that I really am passionate about and wanted to be involved in. And so that's how I came to be the co-chair, co-red list authority for the snake specialist group. And then as the anoline lizard specialist group is getting going again, I'm the red list authority for them as well. And so what that means, of course, is that we're doing these assessments and publishing them as part of the red list updates that are happening. And that's information and data that's really useful for conservation planners and for fellow researchers because it offers us a glimpse into the conservation status of all those species. And it's a lot of work to do these, but it is really valuable and I enjoy it.
Good. Well, I'm glad you're there doing it. So red listing is not something, I mean, I'm interested in the species, but I would not be good at that. And I always found it hard when I was involved, like chairing specialist groups, finding the person to be the red list authority. I always found that kind of the hardest role to fill. But
yeah.
Anyways, glad you enjoy it and glad you're doing it for snakes.
Yeah.
So anyways, we'll start wrapping this one up. But before we do, I like to have all of my guests tell me a good snake story. So, Graham, what's your best snake story?
I have many, but I've got a classic one that is one of my favorites. So this is about the silver boa as well. So the night that we discovered the silver boa back in 2015, again, we were on this remote island searching at night for lizards, and we found one of these boas, and we kind of dropped everything at that point in time and just were like, okay, we're looking for boas now because these look like nothing we've ever seen before. So we ended up finding, I think, 5 in that night, and the boat had dropped us off, and the boat was going to come pick us up at dawn to go back to the main island. And so I said to the team, let's just lay down on the beach and grab an hour or two of sleep before the boat comes, just to get a little rest because we were pretty bushed at that point. So we lay down in the sand, just right on the beach, no tents or anything, and fell asleep pretty quickly. And I remember waking up with a startle because there was something on my face. And it's one of those things where you wake up from in a stupor, and I remember reaching up and thinking there was like an insect or something, but then felt that there was a large mass on top of my face and grabbed it and picked it up. And as I started to come to, realized that it was a boa, that a silver boa had crawled down from one of the trees and across the beach and on top of my head while I was asleep. And I could not believe it. So, why would it have done that? I don't know. I mean, I must have really smelled like a snake at that point because we'd been processing these boas and taking all these data. But I did what anybody else would do. I put it in a snake bag, and tied it up and went back to sleep. Waited until dawn, and then we had one more to measure and then let go. So that to me was just a story that really showed, in my view, I think the silver boas really wanted to make sure that they were discovered.
Yeah, I was going to say, I need to have you teach me how to do that, to have the snake that I'm serving for just come to me.
I know, exactly.
Although I usually work with venomous snakes. I'd prefer not to have them on my face, right. I guess. So anyways, great. Well, I've really enjoyed this conversation and learned quite a bit. So thank you for joining us today. It was great.
Thanks. Enjoyed being here. Appreciate it.
Yeah, and I just wanted to thank the audience and tell everybody to remember snakes are animals too, and it's a privilege to see one in the wild.